Okay, I hope everybody can see my screen. Thank everybody for joining. Looks like we have quite a few people from all over the world. Um, we're going to talk today about two stories, both very recently published. Just last year, we published a paper with BMS involving really big macrocycles, which the story ends up being very physicsy and very much focused on biophysical data, both NMR and X-ray data, starting with a hit from a peptidream screening campaign. So these big 15-mer peptidic macrocycles, they're difficult to manage from a computational perspective, but we've come up with some methods with our colleagues at BMS and work in some other places to do this. So I'll be telling this story, and all these papers that we're going to reference during the webinar are open access, and there's quite a lot of backstory behind this paper. And it's going to talk about another case of lead optimization beginning with a macrocycle, but it's a small macrocycle, a natural product that was then developed into a crop antifungal. And in this case, there's no structure enablement. Rather, it had to be done entirely using ligand-based methods. So without going too much more into the overview here, I'm going to jump right in and talk about the PDL1 story. So what we have here is the initial hit that binds to PDL1, abrogating its ability to bind the PDL1 receptor. And so this is an important cancer target. And so what we had from our colleagues at BMS was a whole lot of structure activity data. I'm showing you just 6 compounds here. And we had big challenges with respect to conformational search. So in trying to approach these kinds of macrocycles, you may or may not have biophysical data. So you can approach these things from an agnostic point of view and do extremely thorough conformational search, which can be productive for these macrocycles, particularly when you have significant amounts of transannular hydrogen bonding. In this particular case, it turns out you don't have a lot of sort of secondary structure, sort of like a protein. And so NMR restraints can help you over the challenge there. In this case, we had both NMR data and we had X-ray data. One of the things we'll talk briefly about is the fact that it's very difficult to correctly fit these kinds of macrocycles, or really any macrocycles, into X-ray data. But if you are able to have some biophysical data, you can turn those into torsional restraints to help you restrict the conformational space of analogs. So you'll see that the initial hit was about 9 nanomolars, 9 nanomolar in a particular assay, and it was improved very substantially to the clinical candidate. So this is, I think, either on its way or in the clinic right now, the 900 compound. So what we wanted to do was to see if we could vastly improve the efficiency of getting from beginning to end, the literal lead compound to the actual clinical candidate, by doing some computational modeling. I want to emphasize this is a retrospective study, so all of these compounds had been made, and then we tried to figure out if we could have done it much better if we applied computational tools. One thing that surprised us in this is the huge importance of ligand strain. So as you look at the structure activity data here, and you look at this simple phenylalanine up here to alanine down here, you lose 1,000-fold in terms of the effective concentration, and that turns out not to be actually primarily an interaction with the protein. It has to do with the conformational preferences, and we'll get into that. So the overall scheme here for doing the lead optimization in this structure-enabled project goes as follows. So starting with the lead compound, there's sort of 2 pathways to winning here. One is we can use NMR restraints, and Luciano Mueller, who runs the NMR group over there at BMS, provided us with, I think it was a lot, it was over 200 NOEs to help constrain the conformation of this peptide. Using our approach, our fourth-gen approach, we were able to come up with a very nice tight solution ensemble for this compound. Turns out there are some X-rays available. This is a publicly available X-ray, 6BV9, of this initial lead compound that came from an academic group. It turns out that when you correctly fit the macrocycle into the X-ray density, it pretty much exactly matches the NMR solution ensemble. And this is something that Ann and I have seen now repeatedly with multiple collaborators on working on these types of macrocycles, that they typically are pre-organized in solution to bind the protein in question. So using either NMR data or X-ray crystallography, we can extract essentially a preferred backbone conformation for the macrocycle, and use that then to help us restrain conformational search on new molecules. So here we're showing peptide 5 from the previous slide. So we have a proposed analog, and we want to generate a conformational ensemble that largely respects this backbone that we derive from our lead compound. From there, we do two things. We do a full conformational search to end up with a reasonably deep exploration of what this compound can do if it is adhering to the conformation that came from our lead compound. We also then do a fully agnostic conformational search that's unrestrained in order to find an estimate of the global minimum energy. Here, we're going to focus on one side of this schema here, where we're using the actual crystal structure, just one crystal structure, though. This is just the 6BD9 structure, where we're going to do docking now using this restricted conformational ensemble. From the docking, we're going to get a predicted bound pose, and from that, we're going to derive the intermolecular enthalpy between the peptidrine, the candidate compound and the protein, and we're also going to derive a bound conformational energy. From the agnostic search, we're going to get the global minimum, and now we're going to be able to derive a score, which is the sort of an enthalpy-focused score based on the intermolecular energy, and then the term in parentheses here is the strain. And the strain, as I mentioned before, turns out to be really important. There is a workflow, and you can read about it in the paper, that's completely ligand-based, where we used no structure information from crystallography, but only used the information from NMR. That doesn't work quite as well as when you have a structure, but it does work very well. But we don't have time to cover both. Okay. One of the things that's difficult about trying to cover this story in about 20 minutes is that before the PDL-1 paper with our colleagues at BMS, there were 8 other papers that really formed the backbone of the technical approach, and we can't go through all of that. So Ann and I started working on the sort of conformer problem with respect to macrocycles in about 2016 or so, and started publishing things in 2017. That led to working with great colleagues at Merck, in particular, Ed Scherer, Misha Rebark, and Qi Gao, where we looked at how to combine a conformational search with NMR data. So I encourage anybody who's interested to go and look at this paper here. We look at NOE data. I think we have some J-coupling data as well, but also RDC data, and we show how to combine those things together in order to effectively search complicated macrocycles, although not as complicated as the ones we're talking about today. We worked with Scott Loki at UCSC to look at these lariat peptides. Really great collaboration with Thomas Williamson, as well as our Merck colleagues, on a particularly interesting variation in terms of conformational space for ariovicidin, which was discussed also in this other paper. We also worked with Thomas and his group at UNCW on rapamycin. This turned out to be really interesting, and I can't tell that story. All through this work, though, what we ended up doing was uncovering more and more and more challenging issues with respect to NMR data and how to actually interpret it and effectively utilize it in doing conformational search. As we worked through those issues with NMR, we started looking at X-ray density fitting as well. And this was primarily with our colleagues at Merck, in particular, Alex Bruckner and Ed and Misha, who I've already mentioned. Turns out that if you want to think about bound ligand strain, the big challenge with complicated molecules is that if you look in the PDB at basically any structure that has a bound macrocycle, the conformation of that macrocycle is grossly correct, but in details really quite wrong, probably 90–95% of the time. If you use the wrong conformation, you end up with ridiculous estimates of ligand strain. So we had to figure out how to refit ligands, both small and large, into X-ray density. That led to a systematic study of conformational strain of macrocyclic peptides that finished sort of Alex's postdoc. This is a neat paper, and one of the things that you find is that these large macrocycles can support and sustain a lot more strain than small molecules can, but not a crazy amount. And it turns out that as we were working through this paper, we noticed that there looked like there was a real pattern in terms of how much strain you could expect a molecule to have based purely on its size. We were then able to derive a model of the relationship between ligand size and bound strain that actually covers everything from regular small molecules through synthetic natural product types of macrocycles like, say, rapamycin or HCD protease inhibitors like vaniprovir, but then all the way up to really big things like we see with these peptidrine molecules and also very large natural product macrocycles. This ends up being really important in thinking about how you do lead optimization for macrocycles because the strain question ends up being a dominant issue. That brings us to the paper that we're primarily talking about here that was done in collaboration with BMS. And the really key players here were Luciano, Christine, and Alex Bruckner. And I enjoyed working with this whole group here. All right, so again, that's 8 papers that support the story that I'm going to tell you. And instead of going through the 8 papers, I'm just going to try to talk through a little bit of what we had to do on this one slide here. So in order to deal with NMR with these kinds of macrocycles, not only do you have to do a deeper form of search than our initial 4th-gen approach that we published in 2017 and 19, we actually implemented a yet deeper search, which helps to elucidate better lower energy conformations that actually fit either X-ray density or NMR constraints better. When you look at the performance of the 4th-gen deep search here in purple, this is on the prime MCS set. This came out of a collaboration between BMS and Schrodinger, where they introduced the prime MCS method. And so what we're showing here is RMSD on the x-axis and cumulative histogram on the y-axis of the ability of any of these methods to find a conformation from an agnostic starting point that's close to what is correct crystallographically. And you can see that the 4th-gen deep method is really substantially better than everything else over here. 4th-gen by itself, without the deep approach, is also better than all these methods, but only by several percentage points rather than sort of 15 to 20, depending on where you are in the curve here. So the deep search mattered a lot. And then there was a lot of hair around dealing with dihedral angle disambiguation and treatment of chemically equivalent protons in NMR data. And all that's talked about in the papers, but it's a little bit technical for such a broad audience as we have today. This paper that we did with Thomas's group uncovers, interestingly, a different conformation of rapamycin that is sort of the minor conformational pool, but this is actually very different than what people had thought for about 30 years. So it's an interesting story. Key to this was, though, all of the work that we did in terms of interpreting NMR restraints correctly. All right. So this is just the underlying technology. So we can do agnostic search, we can do NMR restrained search, and we can take advantage of things like RDC data as well. Okay, so when you're taking advantage of X-ray crystallography data, it gets complicated. So if we look at 2 different structures here of 2 different of these peptides. Peptide 1 is 6PV9 from the PDB, and peptide 57 is a very minor change at 1 position here where we have the serine. If you look at the 2 PDB structures as deposited, the backbone of the macrocycle looks pretty substantially rearranged across many places, and that's puzzling because you wouldn't expect it, and you sort of also wouldn't want it to be true. If you're trying to design things, you don't want to have to deal with massive rearrangements in the backbone. Well, what we did was we refit these macrocycles into the density for these two different structures here. When you do that correctly, what you see is they're actually doing the same thing. So both of these peptides, as you would probably expect based on it being such a very minor structural change, they fit the density on the backbone almost exactly the same way. Even the side chains fit in the same places, roughly speaking. Another huge impact, though, is that when you look at the strain on, in particular, peptide 57 from the original 504Y structure, it's over 100 kcals per mole in its bound configuration compared to its global minimum. If you refit that, it comes down to 7 kcals per mole. That's an absolutely huge difference. There's no way that a ligand like even this size can support a strain of 100 kcals per mole. So getting this right, getting the global energy minimum correct or close to correct, which you can't really prove, but you can try. But getting the fit to the density correct, so you don't have crazy things like cis primary amides or 90-degree amides. Doing that all helps you understand what it costs to bind the protein. In the case of peptide 1 and 6PV9, the structure was less incorrect, but an important issue was they inverted the chirality on one of the pendant side chain here that actually sticks up into solvent. And that has a pretty significant effect on the geometry of the bridge here. After you fix everything, though, again, everything goes and turns into a nice copacetic story between these two different peptides. The strain is very low. So out of our paper where we developed a model of how size of ligand relates to expected strain, what we know is these things are about 130 non-hydrogen atoms. Given that, we expect that the strain should be around 25 or 24 kcals per mole, and the lower and upper bounds are about 14 to 34 kcals per mole. What we find, and this is something that Ann and I have seen many times now in peptidream hits, is that they tend to have very low strain that are below the lower bound of expectations based on what's in the PDB. What that means is that by making changes to this peptide scaffold, you will often hurt yourself by increasing strain drastically. So understanding the distributional expectations and then understanding that our lead compound peptide 1 has extremely low strain tells you that, this is our lead compound down here, that tells you that you need to be mindful that the story is not just going to be the binding footprint between the peptide and the protein. It's actually going to require you to figure out what the strain is and get a pretty good estimate. So again, now to remind you of the schema here, we have our backbone that we got from either NMR or X-ray. We're going to use that to restrain search on candidate molecules. We're going to dock those, we're going to score them, and we're going to estimate strength. Okay, so this is sort of the big slide that tries to combine all of the data. So we looked at 72 analogs here. These were systematically curated, mostly out of the patent that was, I think, from 2014, but also several of the synthetic lead optimization compounds during the project. This is all 72 docked. You can see that they all dock sort of nicely following the same basic backbone structure as the ligand in green, which is the lead compound that we started with. What I'm showing you here in the upper left here is peptide 57 docked into the 6PV9 structure. So this is a non-cognate docking of peptide 57 into the structure that was cognate for peptide 1. And you can see that we get a very accurate docking. The actual RMSD is just 1 angstrom. So this is a huge molecule. Getting that level of accuracy is quite difficult. If we look now at the clinical candidate here in sort of tan, BMT 174900, what we see, I've highlighted the places where there are significant changes on the ligand. Two of the really big ones, and this is something that we see a lot with these sort of surface binding large macrocycles, is that optimizing against the charged residues that you see on protein surfaces ends up being important. So a couple of changes here, the substitution on the indole that gives us a salt bridge with a guanidinium is important at position 10, change to to a lysine like a primary amine over here, gives us an oppositely charged salt bridge on the left-hand side. And then there were some relatively minor changes in the hydrophobic interaction in the shallow groove of the macrocycle. On the right, I'm showing a plot of the experimental binding energy versus the predicted binding energy using the workflow from the previous slide. Compound 1 is our clinical candidate over here. Compound 2 is an extremely close analog, and you can see that these are predicted to be much better in terms of free energy than the vast we over predicted in terms of quality that were not as good as what we would have computed, but we're able to eliminate essentially a fifth of the compounds while retaining the compounds that are quite active. Okay, now back to the story about strain. So this compound here, going from peptide 1 to peptide 5, the only change, it's about 6 atoms here. We're shaving off this phenyl group. We see a few order of magnitude loss of activity in just cutting off that phenyl. What you see over here is where that sits in the binding pocket over here. It turns out that this phenyl makes almost no contact with the protein at all. You would think that with a change in activity this large, normally in normal structure-based drug design, you would figure that this phenyl was fitting into a nice tight hydrophobic pocket, and that the bulk of the loss here is that you're losing interaction footprint with the protein. It's not the case at all. The docking estimated enthalpy changes hardly at all. That's within the noise, really, of these kinds of calculations. But the strain jumps up by nearly 9 kcals per mole. And that's because this phenyl, what it's actually doing to this macrocycle is providing a conformational lock. Essentially, think of like a wrestling move, like an arm bar, where with that there, the victim, in this case, the macrocycle, can't twist out of it. But when you take the phenyl off and you release the arm bar, now the backbone of this macrocycle can do something completely different and find a much lower energy solution conformation. So it's the strain really that's telling most of the story in terms of the activity. And this was actually very surprising to our colleagues at BMS, because they had gone through this whole project and made many, hundreds and hundreds of analogs, but they were looking at their modeling of what they were seeing. And there were a lot of stories that were made up about, oh, we think we're displacing this water, we think we're doing this, we think we're doing that, looking at their internal structures. But I think they're convinced now, we certainly are convinced, that the big story really is macrocyclic strain, the ability of these macrocycles to adopt the conformation that came literally from the lead compound out of the initial screen. This is something that Anne and I have seen now with other colleagues and collaborators that will be hopefully going to be able to publish in the next few months, but there are a lot of things going on with all of that. So I'm going to conclude my piece of the presentation here just by trying to summarize over here that you can use NMR data all by itself. And even in this case of the PDL1 project, it's possible to use a purely ligand-based approach in order to do the lead optimization here. So if we had no structure of PDL1, we could still do this. It's not quite as good as when you have protein structure, so you definitely want a protein structure, but NMR can get you a long way. X-ray data is really helpful. With many of these macrocycles, though, they're really challenging to fit into X-ray density. What we found with most of our industrial collaborators at large pharma is that their internal structures are much better than what we find in the PDB. And not to disparage academics, Anne and I used to be ones for many years, but wow, the X-ray data you find in the PDB, you really need to be careful, because as far as we can tell for macrocycles, even small ones, it's pretty much all wrong. But if you fit these macrocycles into density correctly, you can actually derive good estimates for bound ligand conformational energies. And then also, if you can estimate the global minimum, you can get decent values for strain. Very weirdly, this really surprised us. The strain for small, medium, large, very large molecules follows a size-dependent distribution. This is a really important paper you guys should all look at that we published with our colleagues at Merck in 2023. By this time, I think Alex had moved on to Bristol Myers Squibb. All these are open access. But docking and energetic estimation, as long as you have a good estimate for strain, can really help you, even with macrocycles of this size. So the last piece, I guess, of bad news is that you still have to be able to do agnostic macrocyclic search, even for very large macrocycles, because that's the only way you can get close to a global minimum, and that's the only way you can then estimate what the ligand strain actually is. So all of these papers down here with lots of colleagues led to the ability to actually do this kind of optimization. And again, don't have time to go through any of the details, but all these are open access, and we'd be happy to have follow-up conversations with people who have questions or who are interested. But we really recommend that people spend some time looking at those papers. So now I'm going to hand it over to Anne, who's going to tell you this very different but also very cool story.
Okay, so I'm going to describe an exercise where we're employing a Quanza, our affinity prediction module, to guide synthetic prioritization in a scaffold replacement project where we're beginning with a natural product macrocycle, and we wish to arrive at a non-macrocyclic commercial product. And I want to remind you that this project was done in collaboration with our colleagues at Corteva Agrisciences. So I want to begin by describing how Quanza works. Quanza stands for Quantitative Surface Field Analysis. The input to Quanza is a set of structures. Here we have SMILE strings and their associated activities. We convert 2D to 3D, and then we generate deeply searched conformer pools for each of the training ligands. And then using multiple instance machine learning and a process of iterative refinement, we begin by generating first small cliques and then larger cliques of the training molecules. Here you see a depiction of the surface envelope surrounding the optimal final poses of the training molecules, as well as the induced pocket field. The pocket field is generated from observer points from response functions that are responsive to molecular shape. And you can see the functions depicted over here, hydrogen bond donors and acceptors, both direction and arrangement, and electrostatics through Coulombic field. Then we use the pocket field in a process akin to docking. We can score new molecules and predict both their pose and their affinity. Seen here in cyan is a scaffold that is very different that we're predicting on from these traditional benzodiazepine scaffolds. And this ability to extrapolate to new scaffolds is part of, stems from the fact that Quanza generates models that are physically realistic. Also seen here is this is relative to a pose of one of the training molecules. You see these interaction sticks, and both in magnitude and type, these depict the interaction with the pocket field. And so you can see, for example, this hydrogen bond donor stick, this acceptor stick, and these steric sticks. And in sum, they account for the activity prediction. I want to elaborate on the multiple instance machine learning aspect. These are, for example, 2 steroids training molecules, and you see the observer points surrounding these training molecules. Even though in an optimal click, you do have a single pose per molecule, you also have variant poses for a single molecule. This is important, for example, in this set, an old steroid hormone set, because some of these steroids, if you look at them relative to DHT, for example, their binding modes are flipped left to right and top to bottom. In addition to be able to extrapolate to new scaffolds, Quanza can also accurately predict the affinity of future molecules. This is paper that we published with Stephen Johnson at BMS. Here you see 3 training ligands. These are LFA1 inhibitors. This is their mutual alignment in the training set. What we can do, though, here is we can predict on future molecules that are made 7 months after the last training molecule. And you can see the interaction with the pocket field for this molecule, and that we have a very accurate prediction. We did very well. This is the experimental versus the predicted activities of all 67 future molecules. Interestingly, Stephen Johnson, for those future molecules where FEP was applicable, we showed that Quanza was in fact synergistic with the FEP predictions. These are orthogonal methods of affinity prediction, and we saw that if you average the predictions from each of the methods, that the hybrid method did better, and we showed this for an additional 16 targets in this paper. So for today's talk, this is also a temporal set of molecules, but instead of just having 67 future molecules, now we have over 1,000. So the idea here is that UK2A was a natural product. It is a macrocycle, and it has 4 chiral centers. It inhibits the mitochondrial electron transport system in fungi that attack wheat crops. There is no X-ray crystallographic structure for this target. It's purely ligand-based. Quanza can use crystallographic ligands as input, but in this exercise, this is purely ligand-based. And what we want to do in this thought experiment is we want to apply Quanza in an active learning strategy such that we can efficiently arrive at this commercial product that is synthetic. It doesn't require fermentation. It has only two chiral centers, and it's non-macrocyclic. And in fact, what you're going to see is that we use the first 100 molecules in this data set to build a model, and then we had to select just 100 more to arrive at this desired product. The scheme of this experiment is the following. We're going to take the 100 earliest training molecules, and we had a good range of PIC50s here, which is helpful for Quanza. We're going to take 5 of the most highly active molecules, and we're going to generate a mutual ligand alignment. We're going to use that alignment of the highly active molecules to seed the alignments of the full set of training molecules. We're going to use that Quanza model then to score all 1,000 future molecules. We're then going to get predictions for the affinity and the pose, and we're going to select some of those molecules for synthesis. And so the idea here is that all of these molecules are drawn on a whiteboard, and we're predicting from those that are not yet made in this thought experiment, we're predicting 10 of the most active and 10 of the most informative, which I will describe on the next couple slides. We're then going to take these 20 molecules, we're going to synthesize them, determine their activities, and we're going to refine the initial model of 100 molecules. For each round of refinement, we're adding these 20 molecules to the model. This slide depicts the process, which was objective of choosing which training molecules we would use in a multiple ligand alignment to seed the alignment of the other training molecules. We have a way so we chose UK2A, which was the first molecule. We have a way to choose among the top log of activity those 4 other molecules that were most topologically diverse. We generate the mutual ligand alignment, and we have several hypotheses that come out of the multiple ligand alignment. And from those, we also have an objective criteria for choosing which one. We use this in a high throughput screen for those first 100 molecules, defining those with PIC50s greater than 8.5 as highly active and those with PIC50 less than 7.5 is less active. And we use this in a virtual screen, and we have an ROC area of 0.92. In addition to activity and PROS prediction, Quanza scoring also yields quality metrics. One of these, for example, is the idea of novelty. And this simply is a question of, is the predicted pose of a new molecule covered by the pose of the union of that of the training molecules? And so what I'm showing here on the left is for each round of the automated scoring, prediction, and refinement. These are some of the future molecules that were predicted to be most active in those rounds, but also lie within the scope of the poses of the training molecules. On the right here, I'm giving examples from each round of those molecules that were predicted to be most informative. This idea of what defines an informative molecule is central to this active learning process. We defined it as a molecule, a future molecule that has very high similarity to a high training molecule, but is being predicted with low activity. And so, simply stated, these molecules had information that the model did not yet learn. And you can see from the very beginning that these were non-macrocyclic scaffolds. I'm going to show examples from a few of the rounds of the refinement of the scoring and refinement. On the left here, you can see that in the first round that those predicted to be most active are still macrocycles, unsurprisingly. But on the right, you can see that those that were predicted to be most informative, that they are already non-macrocyclic scaffolds. In the next round, again, we still have macrocyclic scaffolds that are slightly different. In these, what you're seeing in these poses, or in green, is the training pose of UA2A, and in salmon are the poses of those predicted most active. On the right, you can see two examples of those predicted to be most informative in yellow superimposed on the pose of UK2A. Interestingly, we still have 7 out of 10 non-macrocycles over here. And so the model still needs to learn that new scaffold. If we jump ahead to round 3, interestingly, where we've only refined those original 100 molecules, with 60 molecules, 30 predicted to be active, 30 predicted to be informative, the model has already learned that the non-macrocyclic scaffold is inactive molecules. And those, we still continue to have non-macrocycles in the informative molecules. By round 4, we're continuing to see non-macrocycles and those predicted to be most active. Interestingly, that we notice as well, if you look at the right-hand side of these overlays, that hydrophobic shape is dominating the potency of these molecules. And we have seen this in with other targets as well. This is important because other machine learning methods can overweight polar interactions, but in our experience, the hydrophobic interactions tend to dominate the activities of many sets of molecules. By round 5, we have chosen, there's 7 out of 10 macrocycles that are predicted to be most active. And we have in fact chosen the desired commercial product, FPX, as one of those predicted to be most active. You can see the interaction sticks with the pocket field here, where these 2 hydrophobic pockets occupied by these fluorophenyl rings contribute greatly to the scoring of the predicted activity. We wanted to ask, we were very happy that in the rounds that we increased our predictions of FPX as we proceeded through the automatic rounds of prediction, but we also wanted to look at the entire set of 1,000 future molecules. So on the right are the graphs from each of the rounds, the experimental versus the predicted activities. There were very few, even in the early rounds, there were very few over predictions by Quanza in the future set. This diagonal line is perfect prediction, and the two surrounding lines are plus and minus 2 kcals per mole. The red triangle shows that there were a significant number of under predictions in the early rounds, but as you add the 20 molecules to the training set in each subsequent round, you can see that the under predictions decrease and decrease, and that your accuracy is moving within those 2 kcal per mole areas. If we quantitatively looked at it, beginning in round 0, we had 66% within 2 kcals per mole, but 91% by round 5. Within 1 kcal per mole, 36% was increased to 61%. We then wanted to ask another question. In the preceding thought experiment, the molecules had been drawn for us, and we were choosing from among them to refine the Quanza model. In this experiment, we wanted to generate synthetic ideas on our own. So we begin again first with the same first 100 molecules and our 5-molecule binding hypothesis. We then trim the pendant functions from the scaffold that we wish to replace, and we use the left-hand moiety that doesn't vary as a positional constraint, using this as a target in a virtual screen of the 3,000 compound enamine collection. Shown here are 2 of the scaffolds that resulted from that screen. On the right-hand side, in a chemically compatible fashion, we're going to take the pendant functions from one of these hypothesis molecules, and we're going to cross them over onto the new scaffolds. And you can see, if you superimpose in red here in 2D, is the fluoropicoxamide desired commercial product. And you can see how closely these molecules come to fluoropicoxamide just from that first round of screening. So in conclusion, Quanza is an affinity prediction method that yields physically realistic models. The models can be purely ligand-based. They can be iteratively refined, and through an active learning strategy, rapid introductions of novel structural features was possible. And through the addition of only 100 well-chosen molecules, we were able to successfully identify the commercial product. Key to the active learning strategy was the well-defined notion of what makes a molecule highly informative. And that is, it had high similarity to a known highly active molecule, but was possibly anonymously being predicted to have low activities. And finally, synthetic ideas can be rapidly generated through scaffold similarity screening coupled with pendant group swapping. Over here on the right are some of our publications that contributed to this work. I'd like to thank our key collaborators at Corteva, as well as Stephen Johnson at BMS, who was a co-author both on our eSIM and the LFA1 Quanza paper. Also like to thank Pat Walters at Relays, who was an early adopter of Quanza and the predecessor, QMOD. He was also a co-author on these papers with us. And with that, I'll hand it back to Ajay.
Yeah, so thank you. That was great. I wanted to also thank our colleagues again at BMS and Merck with respect to the macrocyclic work that we've done. So Alex Bruckner, in particular, Luciano Mueller and Christine George were just amazing collaborators. And at Merck, we've published, I think, 6 papers now with Ed and Misha and Chi and Charles Lesberg. So it's been huge to be able to work with people at both big pharma and then also a smaller pharma at Aperture, and also with Thomas Williamson's group at UNCW, where we did the rapamycin work. We learned a huge amount about macrocycles and NMR. We keep learning new things about macrocycles and NMR. We also want to thank Matt Siegel and Peter Hunt at Optibrium, who have been wonderful colleagues, and it's been great to become part of the big family at Optibrium rather than it just being, Anne and myself at BioPharmics. I want to remind people that the platform overall consists of 5 different tools. The conformer search that we talked about is in the tools module, and this contains everything to do NMR restrained deep conformer search, and is also highly effective with non-macrocycles. It's not template based, doesn't require libraries, it generalizes to new chemistry. It's a very different approach. In docking, most of you are probably aware that we've been doing docking for longer than just about anybody in the world, in terms of truly class-leading flexible molecular docking. Recently got into the X-ray density fitting business, and that's been interesting because we learned a lot about things like ligand strain, and also about the fact that the best way to fit things into density is not to pretend that atoms are wiggling individually, but instead we fit conformational ensembles of ligands into X-ray density. The similarity approaches are in the similarity module. All of that underpins the Quanza work as well. So the mathematics and the pose and conformational optimization work that is in the eSIM, the similarity module, all of that drives what's in Quanza, what Anne talked about, which is the affinity module. Okay, so if you go to the last slide, Anne. Okay, this is just pictorially reminding people of all the different bits and pieces there. And now we'll be able to answer a few questions. I'm going to cover sort of the questions. I'll read the ones that are relevant to Anne's half of the presentation first, since that's fresher in our minds. Let's see. Okay, so first question for Anne would be how many molecules and what sort of assay and data and activity range is required to build a Quanza model?
So Quanza models can be built with as few as 20 molecules if you have enough structural information. So it's helpful if you have scaffold diversity. It's also helpful if you have a few or many logs of activity data range. And the types of, did you ask about the types of assay values as well?
Yeah.
Yes, so you can have, clearly, if you have biochemical data, that's great. But you can also have qualitative data. For example, you might have just 3 bins of where you say something is largely inactive, or it's active, or it's highly active, in which case you can use surrogate PKD values. For example, you may want to say, you can use inequalities and say this group is greater than 8, this group is 6, this group is less than 4. You can also use surrogate PKD values for things such as percent lethality, and that also works very well.
Right, good. You also covered another question then about whether we can use phenotypic or qualitative data, and so thank you for that. Next question, can you make use of very large data sets like you see in Chembl, where you've got thousands of molecules and maybe, pretty diverse assay conditions?
Yes, so we have done this. We published this with some other targets, especially where we had it was a Schindler data set from Merck in Germany with an FEP set, but we went and we found a large sets of Chembl molecules for those same targets. In those instances, what we like to do is we like to build focused Quanza molecules, models, to make sure that all of the training molecules are in fact competitive. And the way that we do that is you can use eSIM similarity to filter the set down and whittle it down to, let's say, 100 or a couple hundred molecules where you're confident that they're competitive.
Yeah, I just want to add to that for a second that if you really want to build a huge Quanza model, even if you have solid data that's all from the same lab, you usually want to start with a smaller model, 100 or 200 compounds, and then incrementally refine the model to add things to it. We've been very successful with that kind of process. There are a couple of questions that are very general about the Quanza method and whether it's similar to, this, that, or the other QSAR approach. And I'm going to answer that one just by saying that on the slide where Ian showed you the underlying publications for Quanza, they go back 30 years to when we invented multiple instance machine learning at Eris Pharmaceutical. And the short answer to your question is that no, Quanza is not similar to any other method of QSAR that's ever been published. There are a couple things that are very different. One is that it uses this multiple instance machine learning idea, which we also use actually in our docking scoring function estimation. But it is a very different way of thinking about machine learning. It turns out to be technically very complicated, which is why nobody outside of CS does that. So in the computer-aided drug design field, really we're the only ones who do that. The second thing that makes Quanza very, very, very different is, and Ann mentioned this, that it's able to model the detailed shape of the hydrophobic surfaces of compounds. And so when you're looking at methyl ethyl propyl phenyl kinds of modifications to a scaffold that lead to big differences in activity, Quanza is able to construct a physical pocket that actually models that activity pattern really effectively. And we're not aware of any other method that can do that. And the last thing, of course, is that the Quanza model itself is something that you can fit ligands into. So you predict pose, you predict activity, and you get metrics of quality. So all of those things make it very, very different than other approaches out there. So the last question for Ann, how large a macrocycle is tractable using the Quanza methodology? The ones that we saw in the Corteva application were quite small.
Yes. So Force Gen in general, Ajay had shown how well that we perform with, for example, Force Gen deep search on macrocycles. And what we found in all of our studies is that those with rotatable bonds up to 21 to 23 are pretty much behave like small molecules with our tools. And so for Quanza, I would say for now, macrocycles up to 21 to 23 rotatable bonds.
Yeah, so if you look in the upper left on this slide here, you see Veniprevir, which is an HCV protease inhibitor that came from Merck. It is right in that range. So it's about, I think it's 21 total rotatable bonds inside and outside of the macrocycle. And anything up to about that size and complexity basically just totally works. Just run Force Gen for conformational search. You can run it through eSIM, you can run it through docking, and you can run it through Quanza, and you're going to see the kind of performance that you get with what people would call normal small molecules. As you get into the things that I talked about, the peptidreams, sort of 15 more peptidic macrocycles, that's when you really, really pray that somebody can help you out with some NMR data or some biophysical data from crystallography, which is a good segue into the questions now about the larger macrocycles that I talked about. Question one is how large a macrocycle can be handled without any biophysical data? Sort of a two-pronged answer, and already answered half of it, which is 21 to 23 total molecular flexibility inside and outside the macrocycle, you can just do, and that's fine. For big macrocycles, if there is a pattern of transannular hydrogen bonds across the macrocyclic ring, then even if you don't have biophysical data, very often the deep Force Gen search approach is able to get you where you need to go, and it's quite obvious what the correct structure is, and you can do the kinds of things that I demonstrated without any NMR data or any X-ray data. That, in our hands anyway, we've seen that less often with screening hits that come out of these DNA sort of RNA platforms that generate these interesting macrocyclic hits. We generally see fewer cases with strong transannular hydrogen bonding, so you really do want to have NMR data or crystallographic data. Okay, we have another question here. How do you determine which part of the macrocycle strain is coming from? So people tend to always ask this, and it turns out there's no good way to answer that. So if you have a part of the macrocycle, like a substituent that's generating strain, what ends up happening is that that gets distributed across the entire macrocycle. And it may be the case that changing another substituent somewhere else would relieve that strain. So it's something you want to know. What do I have to change to solve the strain problem? But it's not a particularly well-formed question because there's always multiple solutions. You might be able to make a bridged ring. You might be able to change another position on the macrocycle. So attribution of strain to this piece or that piece, it's not something you can compute, but I don't even think it's a thing. I think that that's not the right question to ask. Let's see. We have I think time for one more question or maybe two. Why is refitting a macrocycle into X-ray density important? Isn't it just enough to get an idea of the binding mode of the molecule? And I think that the big central issue there is that if you fit things correctly into density, you can get a really good idea of what the bound strain is. If that turns out to be extremely low relative to the expectations of how much strain a ligand like that can, should, or we expect to bear, that means that as you make changes to the scaffold or to the substituents on the scaffold, you might be breaking the molecule by changing the strain a great deal by moving the global minimum. So if you fit correctly, you can get an idea of whether rigidification strategies will help you or whether you need to work on your binding footprint with the protein while not screwing up your global free energy minimum. And one last question. You seem to rely on force field calculations a lot. Have you verified the energy calculations, especially with respect to strain, using DFT or other methods? The answer is we've done probably more than anybody else, and you should look at the paper on distributional strain modeling that we published with our Merck colleagues in 2023. It turns out to be a very complicated answer, so I just recommend that you go and read that paper. And I think at this point we are out of time for questions, but thank you everybody for attending.